Alright, so this is actually an introductory class of immune system or immunology, clear? Now, today we are going to start a lecture series on immunology, clear? Now, before we start the very intricate concepts of immunology, we must understand that immunology, this word has come actually from immunitas, okay? It has actually come from immunitas. Now, what do you mean by this immunitas? You know, it has actually come from this word, immunitas. You know what do you mean by this word? Now listen, actually this word means immunity, okay? Now listen, this word was actually first introduced or was first used in Roman Empire, okay? It was used in Roman Empire because those people who actually work under Roman Empire, when they were actually doing something bad or something like that, okay, which is harmful for people, then they were actually given punishments, but they were also given some immunity. That means they can actually get away with very big charges with small amount of money or something like that, okay? This is immunitas, okay? So historically from this word immunity has come, clear? Historically from this word only immunity has come. So from the ancient time, this word immunity means body is, or so historically this word immunity means protection, clear? Means protection. Now listen with all your attention. All the cells which are actively helping us, in our protection, all the cells and all the mechanisms which are actively helping us in our mechanism and protection, in our mechanism of protection, these cells and all these things together is called immune system. It's called, these all things taken together is called immune system, okay? Now listen with all your attention. Collective and coordinated, the collective and coordinated response of immune system, okay? The collective and coordinated response of immune system towards a foreign substance, okay, is called immune response, clear? So what is the real meaning of immune response? Now you know, okay? Immune response, clear? Okay, so the collective and coordinated response of immune system towards a foreign substance is called immune response, clear? So you now know what is immune response, what is immune system, and what is immunity, right? Now the evolution of immunology as a science actually dates back to a time where a person was experimenting with something called cowpox, and smallpox, and the person's name was Edward Jenner, okay? And the person's name was Edward Jenner. What did Edward Jenner saw was very interesting. You know what did he saw? Edward Jenner was actually an English physician, okay? Now what Edward Jenner was actually observing was very important for us to understand. Why? Because he actually observed that those milkmaids who actually recovered from a disease known as cowpox never contracted the disease again, clear? Never contracted the disease again. So then what he did was he actually took some cowpox serum or something like that from a victim, and then he injected a small amount of that into an 8-year-old boy, clear? And very interesting thing happened. You know what is that? That boy never contracted a disease known as smallpox. So actually what he understood that this boy was actually now immune to smallpox because he was injected with cowpox materials. now we know that what Edward Jenner was acting, or sorry, now we know that what Edward Jenner was working on is known as vaccine, right? We now know the importance of vaccine in this era of science, right? Now this the vaccine, this word actually has come from vaccinus. This word has actually come from vaccinus, and vaccinus means cows. Clear? Means cows. Okay? This word vaccine has come from vaccinus, which means cows. Okay? Edward Jenner published his paper on vaccination in 1798. Clear? And this was a landmark. Okay? And he was actually behind, the development of vaccine for the first time. Clear? And using his ideas and his own ideas, actually Louis Pasteur again after some years developed a vaccine. Okay, now we all know, for rabies, right? So this is the history of, brief history of immunology. Okay. Now we will be talking about the different kinds of immunity. Clear? This is where we will actually the main thing. Okay, types of immunity. Now listen, very interesting part you need to understand here is this, that there are actually two types of immunity. Clear? In our body, two types of immunity are working simultaneously. Clear? And both the branches are interlinked. And both the branches are interlinked. Clear? Both the branches are interlinked. Now, what are the two types of immunity? Number one is innate, and number two is adaptive. Clear? Number one is innate, number two is adaptive immune system. Okay, that is innate immunity and adaptive immunity. Clear? Now, why we differentiate immunity into two types, that is innate immunity and adaptive immunity. Now listen with all your attention. A very interesting thing you need to understand about these two, that is a striking difference between adaptive immunity. The striking difference between the adaptive immunity or adaptive branch of the immunity and the innate branch of the immunity is this. Do you know what is the basic difference between innate immunity and adaptive immunity? Now listen, the basic difference between this innate immunity and adaptive immunity is this. Innate immunity is immediate. Innate immunity starts with I, and that's why it is immediate. Clear? It is immediate. Okay? It is immediate in nature. That means whenever a bacterial virus enters your body, then immediately your body will start acting against the virus or bacteria, right? Now this is actually innate immune system which is activated. That means whenever a bacteria or virus, suppose this is a bacteria, which is entered your body, it will be at first attacked by innate immunity because it is immediate, and then it will be attacked by adaptive immunity because it is afterwards. A for afterwards. That means after immediate response, late response will come. Okay, and the late response is much more vigorous and much more helpful and beneficial for us. Clear? Now today we will be only discussing this innate immune system or innate immunity. Okay, we will have an series of lecture on innate immunity. Clear? Then we will be discussing adaptive immunity. Clear? Now listen with all your attention. So if we actually try to differentiate between innate immunity and adaptive immunity, then suppose this is a scale. Okay, now listen, suppose this is the event horizon. That means this is the time when infection occurs. Okay, suppose this is the time when infection is actually occurring. Clear? Clear? Now a very interesting point you need to understand is this is the starting point. Okay, this is the starting point. Now listen, within hours we will see that innate immune system, innate immunity is activated. Clear? Is activated. Okay, but after some days or hours, okay, after many days or hours, suppose after 5 to 6 hours, 5 to 6 hours or 2 days, up to 2 days. Now listen, this is the time period when adaptive immunity is working. So now you must understand, that means whenever your immune system is activated, now listen with all your attention, where your immune system is activated, at first innate immune system is activated, then adaptive, right? Now listen with all your attention. A very interesting part you need to understand here is this, that innate immune system is the first branch of the immune system which is activated. Now a very interesting part is this, innate immune system actually activates adaptive immune system, and adaptive immune system will take at least few hours to 2 days, up to 2 days for getting activated properly. Clear? For its proper activation it needs days. Clear? But once it is activated, it will resolve the problem within few hours. Clear? Now listen with all your attention. From my own personal experience and all these things, I can tell you that in most of the infections, our innate immune system fails miserably. Suppose if you have scabies, okay, your innate immune system then and there fails. Clear? And adaptive immune system comes into action. Now listen with all your attention. Many infectious agents, pathogens, have actually developed a mechanism to evade our innate as well as adaptive immune system. They can actually evade our innate as well as adaptive immune system. Yes, they can actually do that. Clear? Then why we will have infections? Why we will have coronavirus if we have got very good adaptive and innate immune systems? Now listen, they are very good, no doubt about this, but most of the times they fail because we have in ourselves deficiencies. We have vitamin C deficiency, we have zinc deficiency, we have sodium deficiency, we don't know, right? And these deficiencies actually, you need to understand, these deficiencies are very big problem for our innate or adaptive immune system, because many proteins need activation, and for this activation they need phosphate, they need zinc, they need copper, they need magnesium. Now if you have all the things available to your immune system, it can actually work well. Okay, what is the right now many people are getting affected by coronavirus, and they are actually okay with it, because suppose if you catch coronavirus, if you get a coronavirus infection, what will happen? Suppose if you are a asymptomatic patient, what will happen to you? You will go to a nursing home or your hospital. There you will be given vitamin C and zinc tablets. That's it. Why? Because these actually boost your innate immune as well as adaptive immune system. That's it. They are actually depending on the doctors are actually depending on your immune system. They are actually depending on these two immunities. That's it. They are actually boosting these immunities so that you can work well, or you can actually fight well with this coronavirus. That's it. Clear? Actually, you must understand that asymptomatic patients do have antibodies against coronaviruses. Now, if I ask you a very tricky question, that is, which one is the older immune system, innate or adaptive? What will be your answer? Which one is the older? That means, suppose if I tell you which immune system is present in invertebrates. Now when I talk about invertebrates, you start thinking about mollusks, right? Phylum Mollusca. Suppose if I am talking about sponges, that sponges also have immune system? Yes. Now can you tell me that invertebrates like sponges do have immune system? What we will be calling that immune system, innate or adaptive? Now listen, innate immune system is actually the oldest immune system. Clear? They are present even in sponges, and you will be astonished to know that plants also do have some cells which behave like immune system cells. Clear? You already know that plants actually produce many things, many products like phytoalexins and all these things, which actually can kill a microbe. Okay, but plants nowadays, we can see that plants do have cells which actually attack the microbes. Clear? Now, so innate immune system, that's why is the oldest immune system, is the oldest immune system. Clear? Is the oldest immune system. This is the first thing you need to understand. It's the oldest and it is widespread. That means even sponges have, innate immune system. Clear? So now if I divide, suppose this is innate immune system. Suppose this is innate immune system and this is adaptive. Suppose this is innate immune system and this is adaptive. Clear? So this is innate immune system and this is adaptive immune system. Now listen. So suppose this is invertebrates, okay, and this is vertebrate. Now, can you tell me that innate immune system is present in invertebrates, right, as well as vertebrates? Innate immune system is absent in adaptive immune, in invertebrates, adaptive immune system is absent, but in vertebrates, adaptive immune system is present. Clear? Okay. Now, a very interesting part, I have not drawn this just because of taking these all, okay, but I will be asking you a very important question. Can you tell me which invertebrate actually have innate immune system? Now listen, innate immune system consists of many types of cells, that is phagocytes and NK cells specifically, okay. Can you tell me in which invertebrate NK cells are present or natural killer cells are present? Now listen, in annelids, NK cells are present actually. Clear? In annelids, NK cells are present. Clear? Now, starting from elasmobranch, that is shark, teleos, shark and all these things, that is those fishes which are present, in marine environment, those are called elasmobranch, okay. Starting from elasmobranch to teleost are those fishes which are present in, sweet water or normal water, not marine water. From elasmobranch to teleost, amphibia, reptiles and mammals, all this, okay. Now listen, very interesting point you need to understand is this, all of them actually have both adaptive as well as, innate immunity. Clear? But now can you tell me that in which, of these lymph nodes are present? Now listen. Clear? Both aves and mammals have lymph nodes. Clear? Okay. So this is very important for you to understand. Okay. So that's why innate immunity is the oldest immune system. Why? Because it is present in, older versions of, or older, kingdoms of, older, phylum of animal kingdom. Okay. Clear? Now we will have a comparative, difference between innate and adaptive immunity. Okay. We will have a comparative difference between innate and adaptive immunity. At first, we will be talking about the cells which are present in innate and adaptive immunity. Clear? Now listen. In innate immunity, you will find cells like NK cells, phagocytes like neutrophils and macrophage. Now listen with all your attention. It is better to write macrophage here, okay? Why? Because macrophage is a bridge between innate and as well as adaptive immunity. Clear? Now the special cells which are especially a part of adaptive immune system only, they are called lymphocytes. Now, why they are called lymphocytes? This is because of the fact that during development of blood cells, they have come from a cell called lymphoid progenitor cell. Clear? They have come from a cell called lymphoid progenitor cell, and these cells always give rise to lymphocytes. Now, lymphocytes are of two types, you already know, that is T lymphocyte and B lymphocyte. Now listen with all your attention. A very interesting part you need to understand is this: for the activation of your innate and adaptive immunity, how this thing works. Now listen, why I was calling this macrophage a bridge, right? Now listen, whenever, suppose this is a bacteria has entered your body with its nonsense, idiotic body surface, which are loaded with peptidoglycan and all these things. Now listen, whenever they are entering our body, NK cells, macrophage, all will now interact with this bacteria, and these cells will now start secreting some chemicals known as cytokines. This is the basic of immune system you must understand before proceeding. Now listen with all your attention. These cytokines now attract macrophage to the site of action. Clear? And now when macrophage comes into the site of action, clear? Now listen, when macrophage comes into the site of action, they are stimulated, okay? And after this stimulation, they go to activate. They now will be going to a proper place, that is bone marrow. They will be going either to bone marrow or thymus. Clear? So they will be going to bone marrow and thymus. Now listen with all your attention. Now they will activate in the bone marrow and thymus. There are T cells, okay? There are T cells. Now they will activate T cells. Clear? Now they will activate T cells, and these T cells will now activate B cells. Clear? And these B cells will now become, after activation, they will become plasma cell. Clear? They will become plasma cells, and these plasma cells will now secrete antibodies, and these antibodies will now act on these, these antibodies will now, suppose this is, bacteria. So now these antibodies will act on these bacteria, and this is the FC portion of this, antibody. And now listen what will happen. This FC portion now will be, this FC portion now will bind with an FC portion receptor. This is FC receptor. Will now bind with FC receptor. Clear? Will now bind with FC receptor. And what will happen is this, after binding with this FC receptor, what will happen is this cell is called NK cell. And these NK cells will now be activated, and they will kill this bacteria. Clear? And also B cells and T cells also will attack this bacteria, and they will try to kill it. That's it. Now listen, binding of this antibody with this bacteria, okay, is called opsonization. Is opsonization, okay? This binding and this whole, FC interaction with NK cell receptors, FC receptor, this is called opsonization. This is called opsonization. Clear? Okay? This is called opsonization. So this is the, basic of your immune system. This is how your immune system works. That's it. In a single shot, if you want to understand how your immune system works, this is the best thing you should understand right now. Clear? But today we will only be discussing about innate immune system, nothing else. Now there are some actually, blood proteins. There are some blood proteins. Associated with innate as well as adaptive immune response. Okay, these blood proteins are called, in case of innate immune system, this blood protein is called complement system. In case of adaptive immune system, this is called antibody. Clear? That means B cells were producing antibody. Now who actually produces complement proteins? Okay, I have written complement system. Okay, it will be complement proteins. Okay, now listen. Now who is actually producing these complement proteins? This is the first question you should understand. Now listen, liver is actually producing these complement proteins. Clear? Liver is the main center from where complement proteins are produced. Clear? Now another thing which makes up innate as well as adaptive immune system is components, not cells, components. One of the best component of your innate immune system is nothing but your skin, which acts as a physical barrier. Now listen, anyone can understand that our skin is a barrier, right, to microbes. But how this barrier works, nobody knows. Why? Because before 2000, 2001 and 2002, we also didn't know how this, physical barrier works. Now we know that TLR signaling and all these things is a part of innate immune system. Clear? Now the skin, which is acting as a physical barrier, and other, components like antimicrobial proteins. Clear? Antimicrobial proteins. Antimicrobial proteins. They are the components of innate immune system. Clear? Now, what our adaptive immune system is offering right now? Okay, our adaptive immune system is actually offering us only antibodies, nothing else. They are offering us antibodies. That's it. Plus lymphocytes. Clear? Okay. So our discussion on innate immune system actually is never going to stop like this. Okay, we'll talk about NK cells, phagocytes, and neutrophil. We'll have a separate video on neutrophil. We'll have a separate video on NK cell and how neutrophil and phagocytes actually helps in phagocytosis and what is the exact process of molecular mechanism of phagocytosis. We'll talk about that later. What are complement proteins and how they work on bacteria and viruses? We'll talk about that. Now, how skin, that is physical barrier, that is TLR signaling, is working on your cells. We'll talk about that, don't worry. But another thing we must right now discuss, that is characteristics of innate as well as adaptive immune system. Clear? Now listen with all your attention. If we talk about characteristics of innate as well as adaptive immune system, if we talk about the characteristics of adaptive as well as innate immune system, we must understand that the most important or striking character about innate and adaptive immune system is that innate immune system does not have a memory. Clear? Does not have a memory. Remember this, they do not have any memory. That means suppose a bacteria is entering today and is trying to rupture your skin, okay, and your innate immune system kills it. Now the same bacteria, if it attacks again, they will actually try the same mechanism. Suppose you have a girlfriend or boyfriend, okay, and he or she is cheating on you and you are forgiving every day. That means what? That means you are innate. Clear? You know that he or she will be cheating, so you are actually immune to that, right? Now listen, this also happens in case of innate immune system, but in case of adaptive immune system, memory is a very important component. Memory is a very important component. That means what? That means when a bacteria is trying to enter your mucosal epithelium or something like that, then your body has actually finished the bacteria. Now, in case of adaptive immune system, what will happen? If the same bacteria tries to attack you again, then body will respond with proper equipments now in a short time. That means whenever your body is facing similar kinds of molecular patterns or pathogens, then your body will be better prepared when adaptive immunity comes into action rather than innate immune system. That is the importance of memory. Clear? Suppose a terrorist is entering your city, okay? Suppose a group of terrorists are entering your city. So at first police will respond, right? Clear? At first police will respond, and if police system fails, police will respond, and then afterwards what will happen? Army will come, right? Now listen with all your attention. Suppose just after 2 days, suppose after the neutralization of those terrorists, just after 2 days, another group of terrorists now trying to attack your city. Then in this case, police as well as your army is now in action in a few moments because they were already alert at first, right? This is the importance of memory. That means in adaptive immune system you have memory. Therefore, a second attack by the same antigen or same pathogen will be deleterious for that pathogen, because in a fraction of a second or a fraction of a time, that particular thing will be destroyed. That means for the formation of memory you may take 1 month or 1 day or 5 days, whatever it is. But if the same thing, suppose if the same bacteria now tries to attack again, then what will happen is this: it will be destroyed in just 1 minute or 2 minutes or 5 hours or something like that. That means formation of memory is a very basic characteristics of adaptive immune system. Therefore, the same similar kinds of pathogen, whenever trying to enter your body, they will be destroyed in a much lesser time compared to the first attack by the same antigen or pathogen. Clear? Okay. So this is the basic difference between innate and adaptive immune system. Okay. Okay. Now we will be talking about in details the innate immune system. Before we start innate immune system, you must understand a very important thing. Now, before we start discussing innate immune system, you must understand that innate immune system actually have two important, characteristics. Clear? Two very, very important and similar characteristics. That is, listen, the first characteristic of innate immune system you must understand is this, that innate immune system responds similarly to similar pathogens. Now, what do you mean by this? Right? What do you mean by this? Now, listen, the basic thing you need to understand about innate immune system is, suppose a bacteria is trying to enter your body. Suppose this is a gram-negative bacteria. Now listen, there are many types of gram-negative bacteria, right? There is Escherichia coli and all these things. Now listen with all your attention. Suppose a gram-negative bacteria is trying to enter your body, then innate immune system at first will be activated. Now listen, the innate immune system which now will be activated against the gram-negative bacteria, right? Be activated against gram-negative bacteria, right? Now listen, the proteins and all these cells which will neutralize this gram-negative bacteria are cells for all types of gram-negative bacteria, whether it is E. coli or other bacteria. Suppose your body knows this is a gram-negative bacteria. Suppose this is a gram-negative bacteria, this is a gram-negative bacteria, and this is also a gram-negative bacteria. But your innate immune system will work for this and this in a similar way. That means it will employ the similar, proteins which it will employ to kill this negative bacteria as well as this negative bacteria. Clear? Another important thing you must understand is this: in most cases, innate immune system activates adaptive immune system. Clear? Innate immune system activates adaptive immune system in most cases. Now we will be discussing components of innate immune system. So, components of innate immune system. Now listen, the first component you must understand is this. Epithelial barrier. Second component is phagocytes. And the third component is NK cell. Okay, so these are the three main components of innate immune system. Okay, so at first we will be talking about epithelial barrier. Okay, epithelial barrier, what do you mean by this? Now whenever I talk about epithelial barrier, some people actually believe that I am talking about TLR signaling. No, absolutely no. It has nothing to do with TLR signaling. You must understand this first. Now listen, there are some astonishing facts about this epithelial barrier. TLR signaling will come in the phagocyte section. Okay, now listen, whenever I am talking about epithelial barriers, okay, I am actually talking about two things. Whenever I am talking about epithelial barriers, we are talking about two things, that is proteins. Clear? Another thing are cells. Now listen, whenever I am talking about protein, now listen with all your attention. So suppose this is your epithelial barrier, or this is suppose your epithelia. Okay, suppose this is your epithelia. Now listen with all your attention. Suppose this is your epithelia. Okay, now this is nothing but your basement membrane. This is nothing but your basement membrane. Clear? Suppose this is the basement membrane of your epithelia. Why we are talking about basement membrane here? Because epithelial cell require a cementing material to sit, right? These are basement membrane. These epithelial cells are actively secreting proteins, okay? Actively secreting some proteins, okay? And these protein secretion increases during inflammation. During inflammation, this protein secretion increases. Clear? During inflammation, this protein secretion increases. Now, listen. The name of this protein, which is produced by epithelial cells and secreted by them, is defensin. Clear? These are defensins. Okay? Clear? They are known by the name of defensins. Now listen, defensin production and their secretion increases on inflammation. When the body is inflamed, or these cells surfaces are inflamed, then defensin secretion increases. Clear? Now these defensins, you must understand this very basic thing, that the defensins are actually cysteine rich, cysteine rich proteins. These are cysteine rich proteins which are made up of 29 to 34 amino acids. Clear? These are cysteine rich proteins. These are cysteine rich proteins made of 29 to 34 amino acids. Clear? Okay. Now, interesting part you need to understand is this, that our GI tract is also having epithelial cells, right? Our GI tract also is having epithelial cells. Now listen with all your attention. These epithelial cells also secrete defensins like peptides, and these proteins are called cryptocidins. These are called cryptocidins. Okay. So these are the examples. Defensins and cryptocidins. These are few examples of epithelial barrier proteins which are secreted when inflammatory response is going on. Clear? Suppose there is an inflammatory response going on here. Okay. Suppose there is a bacteria. Clear? There is a bacteria. Clear? Now a very interesting part you need to understand is this. Suppose this is your epithelial barrier of your skin, okay, and there is a bacteria which is about to attack your skin. Now listen with all your attention. When this bacteria will attack your skin, and suppose they have actually defeated this defensin and this cryptocidins, okay, suppose, now listen with all your attention, then neutrophils will arrive in the skin, right? Neutrophils, okay, let me draw a neutrophil. Suppose this is your neutrophil. Now a very interesting part you need to understand is this, that when neutrophil arise, scientists actually saw that when neutrophils started arising to the site of inflammation, what happens is this, the concentration of defensin increases. Then why it is happening? Now scientists know that these cells, which are epithelial barrier cells, these cells have receptors for two things which are secreted by neutrophil. What are these? These are TNF, that is tumor necrotic factor, and as well as IL-1 or interleukin 1, which is one of the main, what? IL-1 is nothing but one of the main, what? Inflammatory cytokine. It is called inflammatory cytokine. Inflammatory cytokine. Clear? So these IL-1 and TNF are actually secreted by neutrophils, and therefore they increase the production and secretion of defensins. Clear? Okay. Now we will be talking about the cells which are forming the epithelial barriers, okay, or which are present in the on the epithelial barrier, you know. Clear? Now we will be talking about the cells which make up our epithelial barriers. Okay. Now listen. Suppose this is again our epithelial barrier. Okay. Clear? And suppose these are the cells. Now listen with all your attention. Very interesting part you need to understand is this, that this is your epithelial barriers and they are actually, you know, secreting defensins. Now listen, just beneath this epithelial barrier, okay, in this place, okay, there are some cells which are found. Clear? Suppose these are the cells. Okay. Now you will be astonished to know, you know, what are these? They are not present in your blood, but they are actually, you know, present here. What is this? You know, these are nothing but T cells. Yes, T cells and B cells. Now listen with all your attention. Yes, T cells, B cells are also part of your innate immune system, no doubt. Okay, but a very interesting part you need to understand is this, that these T cells and B cells are actually different from the T cells and B cells which are present and which you will learn in adaptive immune system. These T cells actually are called intra-epithelial T cells. Okay, and these B cells are actually called B1 cells. Clear? These are called T cells, these are called B1 cells. They are actually not T cell and B cell, normal T cell and B cell. Clear? Now, at first we will be talking about B1 cells. Clear? These B1 cells are peculiar because they produce IgM or immunoglobulin M. Clear? Produces IgM, and these IgM produced by these IgM molecules produced by B1 cells, they can now act on LPS layer or lipopolysaccharide layer of bacteria. Suppose this is a bacteria and you have got LPS and they have got LPS, sorry. Now listen, IgM molecules now act on this LPS molecule and can destroy these, you know, cells. Clear? But these intraepithelial cells are really interesting. Why? Because they have a different kind of, you know, thing. Why, you know? Now listen, actually these T cells, these intraepithelial T cells, these intraepithelial T cells, now listen, these intraepithelial cells are very, you know, abnormal cells. Why? Because these cells, these intraepithelial T cells, these IT cells or intraepithelial T cells, they respond to any cell which have CD1 protein, Now this CD1 protein is again, you know, peculiar protein. Why? Because this CD1 protein actually looks like MHC class 1. The CD1, yes, this is very peculiar. The CD1 protein actually looks like MHC class 1. Now listen, that means what? That means they have a beta 2 microglobulin structure, right? Yes, they do have beta 2 microglobulin as well. Now listen, in this fold they will be having what? Antigenic peptide. Now listen, in this fold they are not having antigenic peptide, they are having antigenic glycolipids. Clear? And now listen, any antigenic glycolipids bound to this CD1 protein which looks like MHC class 1 and they have beta 2 microglobulin domain as well, will now activate this intraepithelial T cells, and intraepithelial T cells now will try to activate macrophage like ordinary cells, ordinary T cells. Clear? Okay. Sorry. Unlike ordinary cells, they will now try to activate macrophage. Clear? So these are the cells. These are the first cells of innate immune system. Clear? Now we'll be talking about the phagocytic cell, that is the neutrophil and macrophage and all these things. Okay. Now before we start the phagocytic cells, which are a intrinsic part of your innate immune system, I would like to request everyone that you please go to that video of neutrophil, where I have discussed in details how neutrophils comes into the site of action or site of inflammation from bloodstream. Clear? Because we will be starting from there today. Okay. That means now we will be not talking about phagocytosis. Okay. At first, obviously, we will be talking about phagocytosis today, molecular mechanism. Don't worry about that. We will be talking. But before that, we must talk about that how actually after coming into the site of action, how these phagocytes engulf bacteria or virus. Clear? Engulf bacteria or virus so that they can actually phagocytose them. Clear? So this engulfment process is also very much complex. We will talk about that today. Now we will be talking about the engulfment of pathogens by innate immune cells. Clear? Innate immune cells. Now, what do you mean by innate immune cells? Now listen. Innate immune cell means NK cell. Sorry. Neutrophil and macrophage. Clear? So now listen. You know that our endoplasmic reticulum produces glycoproteins and glycolipids. Our blood antigen, that is A, B and O, these are nothing but glycolipids. These are actually antigens, but they are glycolipid in nature. So suppose this is a bacteria. Clear? And these bacteria have cell wall, right? These bacteria always have cell wall. Now in this cell wall, glycolipids and glycolipids and glycoproteins are natural component. In this cell wall of bacteria, glycolipids and glycoproteins are natural component. They are natural. That means they are occurring in this cell wall naturally. Now listen with all your attention. We also have many glycolipids, right? We also have glycolipids and glycoproteins. Clear? Now a very interesting part to understand is this. Suppose this is mammalian glycolipids and glycoproteins. Clear? Now, so what is the basic difference between bacterial cell wall or bacterial glycolipids and glycoproteins, which are components of their cell wall, and what is the difference between mammalian glycolipids and glycoproteins? Clear? One of the example is our RBC antigen. Now listen with all your attention. A very interesting and contrasting difference is this. Suppose this is a protein. Now listen with all your attention. These proteins are loaded with carbohydrate chains. This is called glycosylation, right? When a protein is loaded with a carbohydrate chain, this process is called glycosylation. Now we already know from our previous video of cell biology, that is glycosylation are of two types, right? That is O-linked glycosylation and N-linked, right? Now listen with all your attention. In case of bacteria, what happens is this: these terminal carbohydrates are very important. Now listen with all your attention. In case of bacteria, the terminal, in case of bacteria, the glycoproteins have terminal carbohydrates known by the name of mannose. That means in bacteria, the glycoproteins, in bacteria, the glycoproteins end with mannose. The glycoprotein ends with mannose, right? They end with mannose. Okay. Now listen with all your attention. In case of humans or mammals in general, glycoprotein ends with terminal. Okay, glycoprotein ends with sialic acid. Clear? Sialic acid. Now, in case of bacteria, glycoprotein ends with fucose. Clear? And in case of glycolipids of mammals, glycolipids of mammals ends with N-acetyl galactosamine. N-acetyl galactosamine. Clear? Now, it is very important for you to understand. Now listen. So, what happens is this: neutrophils and macrophage, they search for these terminal fucose and mannose. Whenever they will get a glycosylated protein, and in the end they will get mannose or fucose, what will happen is this: they will start attacking that cell where these glycoproteins are present. Suppose this is a bacteria, and bacteria is having cell wall. Now listen with all your attention. Bacteria is having cell wall. Now on this cell wall there are some proteins. Okay, on this cell wall there are some proteins present. Now listen with all your attention. There are suppose some proteins present. Okay, and you know why glycosylation is very important for proteins? Because it stabilizes the protein. Now listen with all your attention. Suppose these are proteins present on the bacterial cell membrane, and these proteins are heavily glycosylated. Clear? These proteins are heavily glycosylated. Now listen with all your attention. You must understand this. These proteins are heavily glycosylated. Now listen. So this is your bacteria. Okay. Now listen. Listen with all your attention. These bacteria is loaded with proteins. These blue colored things are proteins, and they are heavily glycosylated. Now listen with all your attention possible. Clear? These are mannose, terminal mannose residue. Now listen. Suppose this is your macrophage. Okay. Now listen. Macrophage have mannose receptor. Clear? Macrophage have mannose receptor. These mannose receptor actually act like lectins. Lectins are what? What are lectins? Lectins are carbohydrate binding proteins. Lectins are carbohydrate binding proteins. Clear? Lectins are carbohydrate binding proteins. Clear? Now listen. When this macrophage will now interact with the terminal mannose of this bacteria, it will be destroyed. Clear? By this macrophage. Okay? Clear? So suppose this is a blood cell. Sorry, so this is a blood vessel. Okay? And suppose this is a new monocyte. Okay? You know how to distinguish between monocytes and all? They have kidney shaped nucleus. Now, when this monocyte comes out of the blood vessel, they become macrophage, right? Now, how will your macrophage distinguish between your cell and a bacteria? Now they will distinguish on the basis of this glycoprotein terminal residues, that is whether mannose is present or not. In your cell, mannose is not present. In your glycoprotein, you also have glycoproteins, bacteria also have glycoproteins. Then how do these macrophage differentiate between your cell and a bacteria? Clear? Now listen, you have terminal sialic acid, right? You have terminal sialic acid and bacteria have terminal mannose. Macrophage have mannose receptor, or which are nothing but lectins. What are lectins? Lectins are carbohydrate binding proteins. Clear? Macrophage also have another thing which are called macrophage also have another type of receptor which are called scavenger receptor. Okay.